Perfect. Sorry for the technical difficulties, but I'll start quickly. So my name is Volha Tryputsen, and I work as a statistician and drug discovery at Janssen. And together with my colleagues Wilson and Kathy, we'll be talking about our shiny tools for immune fitness exploration. So quick disclaimer. That opinions that are expressed in this presentation are ours and do not necessarily represent those of Johnson & Johnson. So today we'll be talking about cancer and immune fitness, and also we'll talk about our package and two shiny applications that we developed internally to answer some of the research questions. So if you stick around, I can guarantee that you'll learn some biology together with some R tools as well. So before we proceed, we'd like to acknowledge and say special thanks to our collaborators on this project who were instrumental in moving this forward. So cancer. We all know that cancer is a leading cause of death worldwide, and it's accounted for about 10 million deaths just last year. So what makes cancer tricky is that cancer cells can actually hide themselves from our own immune system. So when T cells that are a major part of our immune system come around, those are the ones that are responsible for organizing and orchestrating the fight, the killing of cancer cells. They actually cannot see cancer. So to overcome that limitation of our own immune system not being able to detect cancer, scientists engineered the superheroes, bispecific antibodies. And the superheroes that are here on the slide actually have superpowers. So with their two hands, they can target or bind to two targets at the same time. So with their one hand, they can bind to cancer cells. And then with their other hand, they can actually bind to T cells and bring those in the proximity of cancer so they can start actually killing the cancer. So it all sounds great. It's a great concept, but there are a lot of research and questions that need to be answered in order to understand how to do it efficiently. So the first question that scientists had in mind is about T cell composition. So T cell composition or different subpopulation of T cells are different among different people. But most importantly, those are different between healthy and cancer subjects. And to understand this, really instrumental for the scientists. So now, if we understand cancer or T cell composition differences between cancer subjects and healthy subjects, then the second question comes when we bring different conditions, and those could be different bispecific antibodies that we're testing, different doses, or even different amount of cancer cells that we have in a sample. We need to understand how all of those conditions are actually affecting our T cell composition and also T cell activation. And then lastly, we care about all of that with one main goal in mind, that we want to be killing cancer cells. So how are those different conditions actually efficient in killing cancer? So scientists at Janssen are really trying to find answers to all these three questions. And by doing so, they really hoping to put the puzzle together. And that research journey we call immune fitness exploration exercise. It's quite an important exercise because scientists are trying to learn a lot, and the information that they learn will be feed into the future drug development. What we can gain from this exercise, well, we can understand patients who are potentially going to respond to our treatments. We can also identify biomarkers of our immune state. So some immune system can be active and ready to fight, but others can be really exhausted. And then also we can understand the impact that some maybe present or past therapies kind of have, what effect they make on immune fitness, and also how having those prior therapies or current therapies will change the response to certain drugs. And of course, all of this collectively will give us a better picture and knowledge to understand how to create better drugs for future clinical trials. So of course, scientists had tools in place to look at the data that we're generating, but those tools were super labor intensive. There was a lot of conditions, a lot of files, regular stuff, a lot of mapping to be done between files. So all of that was really manual and labor intense. Then there were also some different ways of how the data can be pre-processed, including data transformation and normalization. And we statisticians know how important that is, right? So then also we needed to implement some specific statistical method and some better data visualization tools. So this is how we came along as a team, and we developed certain solutions, but also leveraged some existing ones. So I'll talk about how we found answers to the first question, which is about T cell composition and the difference in T cell composition between healthy subjects and those who have cancer. So scientists study T cell composition with flow cytometry. I'm sure some of you know, but others that don't. Usually what happens is the sample is taken out of the subject blood, and then the sample is being labeled with fluorescence antibodies, and then the sample goes through like this funnel where different subpopulations of cells being sorted together based on certain characteristics that you can learn about from that fluorescence antibody binding. And then what we really get at the end is the count of cells in each subpopulation. And of course, along with counts, we can get proportions of those specific cell population within the whole sample. And really, this is the data that we were dealing with. So for each subject, we had a vector of different counts or different proportions for different T cell subpopulation. And then the question that we have at hand is to compare how that is different between normal and healthy subjects, right? So we can look in the multidimensional space, we can only look at 2D, and of course, the exploration tool that we picked was something that we can use to visualize our multidimensional T cell data in 2D space. So we chose to use Radviz, our package, for several reasons, but one of them selfishly was that we had an author working at Johnson & Johnson, so can ask him as many questions as we want about his package. So in a nutshell, I don't have time to go through the details, but in a nutshell, Radviz is a multi-dimensional data visualization tool, which has this idea of placing each of the T cell subpopulations or our variables of interest on a perimeter of a circle, and then attaching each data point, which is our sample, to those subpopulations, or we call them anchors, with a string. And each string will be proportional to the value of that particular variable for that particular subject. And obviously strings pull with a different strength, right? Because the values are different. So you get something like this for each subject. The position of subject will move, and then essentially skipping through, we can see something like this plot as a result of our Radviz projection, where some subpopulations will be pulling some patients of interest, right, or some clusters of patients. So in this case, we see that CD27 and CD4 are pulling these blue subjects on the side. But remember, our question was not just to look at the data, but also to try to understand what are the differences between healthy and cancer patients, right, in terms of this T cell composition. So what Radviz package also has is a Freeviz function or method, which really kind of serves as your supervised tool, where you can feed in the disease label. In my case, it was healthy and cancer, and you can optimize the projection of your samples with kind of leaving only those variables of interest that have the most separation between the two labels. And if you start with this Radviz plot on your left, then at the end of the Freeviz stepwise approach, you will end up with something like that, where in two different colors, we have two patient populations pulling apart, and you'll see the anchors or the populations of interest that actually are responsible for that separation, right? So that was the data visualization tool that we tried out. So if anyone else is trying that tool within the similar exercise, would like to exchange feedback and collaborate if even input. With that said, I'll pass it to Kathy, who will tackle on the next question of interest and how we develop a solution.
Good afternoon, everybody. My name is Kathy, and I'm a statistical consultant for Open Analytics. And I was collaborating with Janssen in developing the tools to answer this particular question of how T cell composition changes under different conditions. So basically, we developed a Shiny app, which we use to, first of all, compare the treatment and conditions effects on different cell populations that were identified in the facts step of the data analysis. And in the app, we provide an overview of all the gating structures and cell populations that have been identified. Like Volha just mentioned, normalization is an option which is incorporated within the Shiny app, and some predefined methods of normalization are included. To analyze the data, we use dose response fitting on the identified populations, and there's an option to do the fitting on cell populations of interest or all the populations. As a second option, we also try to rank the curves, the dose response curves from these different cell populations, and these two work hand in hand for different cases, as I'll explain a little bit later. Based on this dose response fitting, we can extract certain parameters of interest, such as the EC50 estimates or maximum effect estimates, and these can be compared across different treatment conditions. As a first step, of course, we need some data to analyze. So this is a short, small screenshot of how the app looks like. You're able to upload the raw data directly from the machine, and this helps reduce errors that can be incurred during pre-processing of the data. For example, when you're copying and pasting between software programs. Next to the raw data, we also would like to map some information associated with the samples in the wells. So we have a plate map that you can either upload into the Shiny app, or you can create from the Shiny app itself. And with this, you can enter information on the treatments that have been used, doses that have been used, donor information, and other meta information that can be necessary for downstream analysis. So like I mentioned before, dose response modeling is the first step in trying to characterize our dose response profiles for the different superhero treatments that we have at hand. So within the Shiny app, it's possible to fit several different models for all the different conditions that are available, the different treatments, as well as the different populations that have been identified. Naturally, this can be tedious if you're doing everything manually one by one, but by using a Shiny app, you can actually do all this in one go and have all the results in one step. The app also does some pre-checks on the data. Sometimes you might have single donor information or multiple donor information, and the models fitted are adjusted accordingly depending on how the data looks like. There are also some checks as well, such as the number of unique dose concentrations, and this helps to avoid forcing models on data when you have fewer points, which is overfitting. So like in the previous slide, there were two dose response curves that were plotted, but in the actual data, we had three compounds that we're investigating, but the other compound did not have a full profile enough that a model can be fitted. So in order to combat this, we also incorporated a second analysis method where we rank the compounds by their raw dose response profiles. So the actual method of ranking is not necessarily important, but it's predefined in the app. And based on the method that's selected, you can assign a score to all your compounds, and thereby you can manage to rank the treatments. And in that way, you can better select the better treatments or compounds that you have in your data. And this uses just the raw data from the dose response profiles. And this is basically what our FACS dose response Shiny app does. So I will pass the baton on to Wilson to explain the third research question.
Hi everyone, I'm Wilson. I work in J&J R&D, and I'll be presenting the third part of this project, which is essentially to evaluate the different treatments on the killing of cancer cells. Next slide, please. So the objective here is to compare different treatments or conditions across multiple dimensions. And in this case, we are looking at three different dimensions of factors. The first one is the amount of cell killing, cancer cell killing. The second one is the concentration of the treatment that is used, and also the exposure time, the time of exposure of cancer cells to each treatment. So the question here is to select the optimal treatment given these three factors. And ideally, the best treatment would be one that eliminate all cancer cells, so 100% of cancer cell killing at a very low concentration, and also while requiring a very low, a minimum exposure time to the cancer cells. So with Shiny, we were able to develop the T-cell K2 app, which allow us to integrate multiple different modeling approaches to rank different treatments or conditions based on these three factors. And in addition to that, the app also allows enhanced visualization, 2D and 3D visualization, to better explore the data. Next slide, please. So here on this slide, we have a sneak peek of some of the interfaces of the app. The app has multiple different compartments, but here we just have part of the interface. On the left-hand side, we have dose response profile of the different treatments that we, in this example, would like to rank. And each treatment profile summarizes the activity of each treatment based on the time, the amount of cell killing, and also the concentration. And based on this profile, we are able to rank the different treatments based on characteristics from this dose response profile, which you can see on the bottom left corner. So the bars represent the different, the colored bars represent the different treatments in the order of activity. So from highest to the least active treatment. On the right-hand side, we implement a different approach, which is fitting a dose response model. As my colleague Katie already mentioned, the app also does a check. If there are multiple donors, it adjusts the modeling to account for multiple donors, in that case, mixed effect modeling. And on in the bottom, we provide a summary of the AC estimates, AC50 estimates from this dose response fitting, and the maximum and minimum effects which can be used to compare the different treatments. Next slide, please. So in summary, prior to the Shiny development, we had a very labor-intensive process which was prone to error. But with the help of our Shiny, we were able to develop 3 different apps which allow us to efficiently tackle the questions that scientists wanted to answer in a very efficient way and automated way, which also allows for, it also makes it more user-friendly for the scientists. And I would say that is all. And if there are any questions, please go ahead. Thank you.
Cool.